Hello everyone. This presentation is about Nephrolepis. Nephrolepis is also called sword fern. And Nephrolepis, this is the member of pteridophytes. So first we see classification, division Pteridophyta, class Polypodiopsida, order Polypodiales, family Polypodiaceae, and genus is Nephrolepis. Father of pteridophyte, Edward Klikoski. Father of pteridophyte in India, Professor S.S. Beer. Pteridophyte term coined by Ernest Haeckel. Smallest pteridophyte is Azolla, and Azolla is a aquatic. Tallest pteridophyte or largest is Cythia. Nephrolepis is a common fern which grows in moist, cool and shady places in forest, hills, etc. Nephrolepis exaltata is a common species. Various stages in the life cycle of Nephrolepis are completed in 2 phases. Each phase is known as a generation. The sporophytic generation is the dominant, while the gametophytic generation is an alternate phase. This is the structure in front of you. You can see ramenta on the upper side, ramenta and hydrothode, and rhizome in the lower side. Sporophyte. The sporophytic generation is an asexual, diploid phase. It begins with the zygote after fertilization takes place. The adult is represented by the fern plant. External morphology. The adult sporophyte is a perennial evergreen herb. It is differentiated into true roots, a stem, and leaves. The plant has a well-developed vascular system. The stem is represented by a rhizome and stolons. The main stem is an underground rhizome. It is hard, woody, and dark in color. The rhizome grows obliquely erect and is covered with persistent bases of fallen leaves. It produces green aerial leaves and lateral branches called stolons. The main function of the rhizome is perennation. Stolons are the subaerial lateral branches of the rhizome. These spread in all directions and produce new fern plant through vegetative propagation. Each stolon is slender and cylindrical with a smooth surface. Adventitious roots are known to develop from stolons. Nephrolepis has well-developed adventitious fibrous roots. These are branched and help in fixation and absorption. Fern leaves are large, green and aerial. They develop from the rhizome. Young leaves are delicate, coiled like a spring, and show circinate vernation. These are covered with numerous scales called ramenta. Scales protect the young leaves and fall off as the leaf matures. A fully formed mature leaf is commonly called a fern front. It is up to 5 feet long and unipinnately compound. The front has a long petiole. The main rachis bears numerous leaflets arranged in 2 rows. Each leaflet it has a midrib with many forked lateral veins. Hydrathodes or water stomata are present at the end of lateral veins on the upper surface of each leaflet. These appear as minute spots and help in removing excess of water and salts from the plant by guttation. The exudate salts get deposited on hydrathodes and form white spots. Hence, hydrathodes are called chalk glands. The fern plant is a diploid sporophyte. It shows asexual method of reproduction by spore formation. The spore-producing structures are called sori. These develop on the lower surface of leaflets Leaves with sori are called sporophylls. This is the structure in front of you. You can see in the upper structure, upper surface, vascular bundles, lower surface and margins. Anatomy. The transverse section of a fertile pinna passing through a sorus shows the internal structure of the leaflet as well as that of the sorus. The internal structure of the leaflet is differentiated into upper and lower epidermis, spongy mesophyll and vascular bundles. Both the upper and lower epidermis are single-layered, non-green and covered with cuticle. Stomata are generally present on the lower epidermis only. Mesophyll tissue is spongy in nature. This is composed of thin-walled, green and loosely arranged cells. The vascular bundle of lateral veins are seen in the mesophyll region. Each vascular bundle is concentric and heterocentric. It has xylem cells in the center, which is surrounded by the phloem cells. The gametophyte represents the sexual haploid generation in the fern life cycle. The haploid generation begins after meiosis in diploid spore mother cell. The adult gametophyte is called a fern prothallus. It is produced on moist, cool, and shady places or shady soil. A spore germinate under favorable conditions. This is the structure of prothallus in front of you. The prothallus is very small, delicate, and almost transparent. It is heart-shaped and flat with dorsiventral symmetry. The prothallus has a deep apical notch. An apical cell is situated in the notch. The central part of the prothallus is fleshy and is thick because of a number of cell layers. This is called the cushion region. The peripheral part has only one cell layer and is called the margin. Numerous hair-like rhizoids develop from the lower surface of the posterior margin region. Rhizoids help in fixation and absorption. The cells of a prothallus contain chloroplasts. Therefore, it is photosynthetic and independent. The prothallus produces male sex organs and female sex organs during sexual reproduction. These develop on the lower surface facing the soil. Reproduction. The fern life cycle shows both asexual and sexual reproduction. Asexual reproduction takes place in the sporophyte, while sexual reproduction takes place in the gametophytic generation. Asexual, as stated earlier, this takes place by spore formation. Spores are produced in sporangia, which develop in sori. Each sporangium has a cylinder multicellular stalk and a body. This is called capsule. The capsule is oval or circular and flattened on two lateral sides, like a biconvex lens. The capsule wall is single-layered and composed of thin-walled cells. There is a ring of special cells in the wall that runs around the capsule along its median edge. A major part of the ring is formed by the annulus, in which cells have very thick inner walls. This is the structure. First one is sporangium. Second is sporogenesis. Third is liberation of spores. The remaining part of the ring is called the stomium region. It is formed by large, thin-walled, delicate cells. This ring plays an important role in the liberation of spores. Each sporangium contains 16 diploid spore mother cells. These divide by meiosis and produce 64 haploid spores. Nephrolepis is homosporous. All spores are small and kidney-shaped. The diploid sporophytic generation ends with the meiosis. Liberation of spores. The spores are liberated from the capsule. When the sporangium matures, the sporangial wall breaks open at the region of the stamen and the spores are liberated through the opening. This is caused due to movement of the annulus in hot and dry conditions. These are some questions. Please give the answer by yourself. Spores of pteridophytes are, if all the spores are of same size and shape, the plant is called. Next is seed habit originate in. Sporophytic generation is dominant in. First vascular land plants are. Water is needed for fertilization is. Fossils are those species which are. Telome theory was proposed by. Prothallus occur in.
What?
Rhynia was discovered by. These are the name of scientists. So sexual reproduction, it takes place in the haploid gametophytic generation and involves the formation of specialized male and female sex organs. Antheridium, this is the male sex organ. Antheridia develop on the lower surface of the prothallus in the region of posterior margin among the rhizoids. Each antheridium is sessile. It has a very small, delicate and globular body. The body wall is called jacket. It is composed of only 3 cells: basal, cup cell, middle ring cell and apical cap cell. Antherozoids are produced in each antheridium. These are haploid male gametes. Each one has a spirally coiled body with many flagella. The wall of the mature antheridium breaks open in the presence of water and the antherozoids are liberated on the moist soil. This is the structure of antherozoids. And this one is of archegonia. You can see clearly neck and venter. Neck is the uppermost portion and venter is the lowermost. In neck, M.Sc and B.Sc are found. Only in neck, only neck canal cells are found. In the center, where the neck cells end, in the end, sorry by mistake, in the center, egg cell is found. So archegonium, this is the female sex organ. Archegonia develop towards the lower surface of prothallus in the cushion region. Each archegonium is sessile with a flask-shaped body. It is differentiated into basal globular venter and a tube-shaped neck. The venter is embedded in the tissue of cushion, but the neck projects out towards the soil. It is slightly curved. The venter cavity contains a female gamete called an egg cell and a small venter canal cell. The neck cavity contains one neck canal cell with 2 nuclei. The neck of the mature archegonium opens in presence of water. The venter canal cell and the neck canal cell disintegrate. Their contents form a substance similar to mucilage, which comes out through the open neck. Mucilage contains malic acid, which attracts the antherozoids for fertilization. This phenomenon is called chemotaxis. This movement is due to malic acid, so this is also called chemotactic movement. Fertilization. Many antherozoids may enter the open archegonium. However, only one fertilizes the egg. This results in the formation of a diploid zygote in the venter cavity. Haploid gametophytic generation ends with fertilization. The diploid zygote starts a new sporophytic generation and develops into a fern plant. Alternation of generation. The sporophytic and the gametophytic generations in fern are produced in alternate succession, one after the other. This phenomenon is called alternation of generation. Meiosis and gametic fertilization are the two important events which regulate this phenomenon. As the sporophyte and gametophyte are morphologically dissimilar, it is described as a heteromorphic alternation of generations. This is the schematic diagram of life cycle of Nephrolepis. The sporophytic generation ends when diploid spore mother cells divide by meiosis and produce haploid spores. The spore starts the gametophytic generation. This generation ends when the haploid gametes fuse during fertilization and form a diploid zygote. The zygote again starts a new diploid generation. Alternation of generation is represented schematically in the life cycle of a fern. So this is the summary of this whole chapter. Pteridophytes are vascular cryptogams with a sporophyte-dominated life cycle. The diploid plant body is differentiated into true roots, stem, and leaves. Nephrolepis is a common fern. Sporangia are organized into distinct sori. It is homosporous and produces haploid spores as a result of meiosis in diploid spore mother cells. The gametophyte is totally independent prothallus. Sex organs are multicellular. Antherozoids are multiflagellate. Fertilization is water dependent and takes place inside the archegonium. Meiosis and fertilization are responsible for alternation of diploid and haploid generation in the life cycle. So thank you very much. We will meet in our next lecture. Hello everyone. This presentation is about Nephrolepis. Nephrolepis is also called